Okay, I'm going to talk about the lysogeny and different types of eukaryotic viruses and mix them. So, first, in the viral life cycles, there's the virulent cycle, which is when viruses always lyse and kill the host after infection. And they also have the temperate cycle, which is when viruses establish long term stable relationships, but they are still capable of virulence. And they can enter lysogeny, which means a few viral proteins are produced, and the viral genome is replicated with host chromosomes and passed to daughter cells. A lysogen is a host cell that harbors temperate viruses. And then the life cycle of a temperate phage. Some examples of this are lambda and P1. In lysogeny, genome is either integrated into bacterial chromosome forming a prophage, or in lysogeny, it is maintained by a phage encoded repressor protein. And inactivation of a repressor induces the lytic stage. This is called induction, and it is when viral DNA is excised. Phage, early, middle, and late proteins are produced, and virons are produced, and the host lyses. And then a cell stress, like when a cell is under stress, it induces the lytic pathway, which is the viral repressor protein is broken down by Rec-A protein of the host due to UV and other things. So now I'm going to talk about the viruses of eukaryotes. So some of the major parts of a eukaryote virus are the capsid, the DNA/RNA genome, the infection, takeover of host, assembly, and release. Most of these parts are universal throughout. Eukaryotic viruses are classified by their genomes. So they have dsDNA, ssRNA, and then another rare one is dsRNA. And they have reverse transcriptase, which is RNA vir. Animal viruses often have envelopes, as we talked about earlier, like envelope virus. They have a phospholipid bilayer. And three of the main differences between a eukaryotic virus is that the entire viron enters the animal cell, and the replication genome happens in the nucleus. And they have viroplasms, which are membrane-bound viral factories that form in some eukaryotic cells to increase viron assembly rate and protect from the host defense. Some viral infections in animal cells are studied in a cell culture, which are in formed plaques, and they bind to specific host cell receptors, typically used for cell-to-cell contact or immune function, like the HIV virus. Host cell entry normally occurs by fusion with the cytoplasmic membrane or endocytosis. Uncoding occurs, and this is at the cytoplasmic membrane or the cytoplasm. The viral DNA genomes enter the nucleus. Most viral RNA genomes are replicated or converted to DNA within the nucleocapsid. And then the way animal virons assemble. After genome packaging, many animal viruses are enveloped. And this occurs during the exit through lysis or budding, when the virus picks up part of the cell's cytoplasmic membrane and uses it as part of the envelope. So when animal viron assembly, there's 4 outcomes, which are virulent, latent, persistent, or transformation. So a virulent infection is when the cell lyses the host cell, and the most common type of this one is the flu. A latent infection is when the viral DNA exists as a provirus, but it's not actively reproducing, so the host cell is still unharmed unless it becomes virulent and the pathway is triggered. Some examples of this are herpes simplex or cold sore eruption. Persistent infections or the slow release of virions from a host cell by budding. This does not result in host cell lysis, but the infective cell remains alive and continues to produce viruses. Some examples of this are AIDS or chronic hepatitis. And then transformation is the conversion of a normal cell into a tumor cell. This is like commonly seen when like cancer, how cancer forms. So viral infections of plant cells. Plants viruses share many common traits with the animal viral trait genomes. But three major differences are they have a wide host range, they're not enveloped, and transmission is different. And the plant cell wall prevents endocytosis. So the viruses enter through wounds or penetration by insects, nematodes, or fungi. After the capsid is removed, the genome is replicated and new virions are assembled, and the movement of proteins helps viruses travel through plasmodesmata channels, which can infect the entire plant.
And then this is what
our completed concept map looks like.
Has all the different parts about the virus that we talked about. And that is chapter 5.
And we hope you are educated about viruses now.